In this video, we are solving and understanding the concepts behind question number 4 of GATE Biotechnology 2021 paper. The question is, which one of the following methods is used to test the significance of a predicted phylogeny? Option A, bootstrap. Option B, maximum likelihood. Option C, maximum parsimony. And option D, minimum evolution. Before we answer this, let's try to understand about the methods used for phylogenetic tree construction and evaluation. In this video, we learn about the methods used to construct and evaluate phylogenetic trees. So there are two major types of methods for phylogenetic tree construction, the distance-based and character-based methods. In distance-based methods, we have least square, that uses neighbor joining algorithm, and UPGNA method. Both of these are called minimum evolution methods as they construct a tree that requires minimum number of evolutionary changes. Under character-based method, we have maximum parsimony and maximum likelihood. So the main difference between the distance and the character-based methods is the type of input data used. So for character-based methods, like the name suggests, characters, that is the aligned nucleotide or the protein sequences are directly used to construct the trees. So whereas in distance-based methods, the sequence data, the characters, are transformed into numbers. They're transformed into pairwise distances, that is the measure of dissimilarities between two sequences, like the percentage of positions where the nucleotides don't match, which is given by the number. So this matrix is then used for building the tree. So with all these methods in hands, now how do we choose an appropriate or relevant method to construct trees? So for sequences with very high similarity, maximum parsimony is the best, and for very dissimilar sequences, maximum likelihood is the best, and for moderate levels of similarity, distance-based methods are the fastest and the best. So let's first look into the details of these methods. So first, speaking about character-based methods, it is also called discrete method and is based directly on the sequence characters rather than the pairwise distances like we just saw. So the two most popular character-based methods are maximum parsimony and maximum likelihood. So maximum parsimony predicts the best evolutionary tree that requires the least number of steps to generate the observed variation in sequences. It is best suited for very similar sequences while working with a less number of sequences. But it is time consuming as it tries to examine all possible trees in a tree space. So phylip and PAUP, these are some tools that are used to perform analysis using this method. So to understand the maximum parsimony method, let's try to construct the evolutionary relationship of the great apes, the chimpanzee, the gorilla, the orangutan, and humans. So to construct the phylogenetic tree, we choose the globin pseudogene nucleotide sequences that are conserved across all these organisms. So just looking at these sequences at most sites like here and in here, we could see that the orangutans are different from the rest of the three, and so it would be considered as an outgroup, while the other three taxa, the human, the chimpanzees, and the gorillas, could be placed by any of these combinations. So for further construction, we choose sites, that is the positions along the sequences that are informative enough to explain the divergence of organisms. So shown here are some examples of uninformative sites. So like at this position, all the organisms have a T, which does not help us to group these organisms together. So here is an example of another site where only the humans have a T while the others have a C. So this doesn't help us either because the three organisms have an ancestral allele while the humans have the derived allele. So to build a tree, we need shared derived alleles. it doesn't really help, because when we try to map where the T in the humans evolved, it could be anywhere. It could be evolved here or over here. But that is a known fact that something has diverged from some other organism. But we cannot group the organisms unless we have a shared informative site. So such sites are uninformative. And here is a site that has shared derived alleles. So we could see that gorillas have an ancestral allele T shared with orangutans, while humans and chimpanzees have the derived allele G, and hence they could be grouped together. So the rule of thumb is that for a site to be informative, a character must appear in at least two taxa, and there are at least two characters. So let's see where on the tree this G allele could have evolved. So in this first tree, we can have the G evolved here. So the humans and chimpanzees have a shared derived allele G that evolved from the ancestral allele T at this branch. So this is just one evolutionary change. When we now try to map the G's on the second tree, it has to have evolved twice. So it could have evolved here and here. So it could also have evolved here, which is also possible, but it would have to switch back to a T in the gorillas, which accounts for another change, and that would still be two evolutionary changes. And when we try to map it to the last tree, we have a similar scenario where the change from T to G has happened twice. It has happened here, and the change has again happened here. So in this tree, we have two evolutionary changes as well. So if we look at these trees based on one site in the genome, it could be seen that the first tree is more parsimonious, that is, it requires only one evolutionary change. So a similar thing would be computed for all informative sites in an alignment for the entire genome, and the tree that requires a minimum evolutionary change is considered the best tree and the most plausible suitable evolutionary tree. So this is a simple example of the maximum parsimony method used for phylogenetic tree construction. Now moving on to the next character based method, which is the maximum likelihood method. So the maximum likelihood method presents an additional opportunity to evaluate trees with variations in mutation rates in different lineages. So this method can be used for more diverse sequences that are not very well handled by maximum parsimony methods, because it could find many informative sites. So it uses probability calculations to find the tree that best describes the variation among species. So these are examples of some tools that perform this algorithm for phylogenetic tree construction. So the maximum likelihood estimations are computed based on certain assumptions. So it is assumed that the characters, that is the nucleotides in A, T, G, C, they evolve independently. So each nucleotide would have a different probabilities value, while the mutation rate is uniform across the positions and branches. Although this method is a bit complicated, the most simple explanation is that the product of probabilities of the lineage taking into account the nucleotide at a site and the evolutionary mutation rates, is computed. So since it is assumed that the probability of a nucleotide occurring at a site is independent of the previous node, the conditional probability becomes P of A given B is equal to P of A. For example, here the probability of the occurrence of A given T at the previous node is just the value P of A, because A here is independent of T being here. So the likelihood of the whole tree would be the product of all these values of the transitions, and the tree with the highest likelihood value is the final tree. So here is another example. We consider this particular site, and these are the leaf nodes. The mutation rates along the branches is assumed to be 1 here, so let's not worry about that. So we assign nucleotides to the internal nodes based on the leaf nodes. So here the internal node, the parent node for these two leaf nodes would be T, and here it could be A or G. So here in this example we just assume it to be G, and we can compute the likelihood for this given tree. So the likelihood of this tree would be the probability of the parent node T and the transition here T to G. So the probability of G given T, and here's another transition G to A. So the probability of A given G. So we construct several all possible combinations of trees and calculate the likelihood based on this method, and the best tree is that with the maximum probability. So this method is limited by the fact that it is computationally intensive. So these are some examples of softwares that use maximum likelihood method to compute the phylogenetic tree. So that's it with the character based methods, and now moving on to the distance based algorithms. So these are minimum evolution methods. So let's first look at UPGMA. So UPGMA is a distance based method used to construct trees based on the minimum evolution method. It is abbreviated as unweighted pair group method with arithmetic mean. It assumes a molecular clock. The molecular clock hypothesis states that DNA and protein sequences evolve at a rate that is relatively constant over time and among different organisms. So this is just similar to the mutation rate, that is the T that we just saw in the previous maximum likelihood method. So for example, researchers have used this molecular clock to estimate the age of the common ancestor of humans and chimpanzees. So this method is used to produce a rooted tree and is considered to be the simplest and the fastest method. So it also assumes an ultrametric tree that satisfies the ultrametric condition, in which the distances from the root to every branch tip are equal. So in other words, the two greatest distances must be equal. So UPGMA starts with the matrix of pairwise distances. So each sample is denoted as a node, that is the leaf, and the nearest nodes, that is nodes that are which have closest scores, that is minimum distances, are combined together and a parent node is created. So the edges connecting these nodes are given the same length, so that the depth of the group is d of AB divided by 2. So now these two nodes A and B are merged together in the matrix, and we recompute the distance between K to other nodes until there's just one node left. So for better understanding, let's just look at an example. We have a simple example here where the scores are all the same, so it's just like perfect numbers over here. So we have a distance matrix. The smallest distance here, as we could see, is 10, so which is the distance between the sample D and sample E. So we branch these two. We just lay out the branches D and E, and we give each branch a score that adds up to the total distance of 10. So 10 divided by 2 is 5. So each branch is given the score of 5 and 5. So in the matrix, we then merge these two samples together. So here it's easy to merge because the values are the same. So DE here, so the distance between A to DE, it becomes 26, and B to DE becomes 26 again, and C to DE becomes 16. And again, recomputing the nodes based on their distances, we here 16 is the smallest value. So DE is, this means that DE is closest to C. So we add up branch C here and distribute the scores as 8 plus 8 to add up to 16. So we give this branch 8, and here this whole thing we already have 5. So this has to add up to 8. So we give this internal node, the branch to this internal node, a value of 3. So again, recomputing, we now merge D, E, and C together. So merging it, we have again 26 here, and these 26 is merged here. So now what we have after merging the matrix here, what we have left is AB merged and DEC merged. So merging these values, we just have 26. So the total distance of the whole tree would be would add up to 26. So like you could see here, we just merged the trees. We already had 10, 10, and 8. So now finally the distance between the two clusters AB and DEC is 26, like we said. So it would be 13 for this cluster and 13 for the other cluster. Since we already have 10 here, this branch would be given a score of 3 to add up to 13. So in the other cluster, we already have 5 plus 3, which adds up to 8. And then to finally add up to 13, this value would be 5. So this would be your final scaled phylogenetic tree. That is, the distance indicates the evolutionary rate over here. So the previous example was simple, where the distances were the same number. Here is a more complex example. So we start with grouping D and E again and give 5 plus 5, 10. So while merging the matrix values, since the values here are not the same, we just take an average of 18 and 20, which is 19 here, and 42 for 41 and 43, and similarly for the rest. And then we merge the next closest node, which is C. So DE is closest to C. We add C, and we again distribute the distances such that they add up to the total distance of 19. So 5 plus 4.5 would be 9.5, and 9.5 plus 9.5 would be 19. And we compute the rest of the clusters similarly. So the tree is the same as the previous example, but just the distances were a bit different here to show a complex example. So this is how a tree is computed using UPGMA algorithm. So here are some softwares that use UPGMA algorithm to construct trees. So Mega is the most widely used software. And we already learned that this method is advantageous as it is simple and fast. But they are also limited by the fact that they assume a constant molecular clock, which is not the case for real-time scenarios. So the other distance-based method, like we had seen in the flowchart in the beginning, is the neighbor joining method. So the neighbor joining method is similar to the UPGMA, except that the weights for the branches in a cluster are not equally distributed. So like in the earlier example, we saw like for a distance of 10, we just equally distributed it as 5 and 5 to like both of the branches. So but in this case, we don't have equal weights for the branches, and because this model doesn't work under the assumption that the samples are evolved with a constant rate. So the neighbor joining NJ algorithm allows for unequal rates of evolution so that the branch lengths are proportional to amount of change. So this would give us a scaled phylogenetic tree where a shorter branch signifies a shorter evolution time, and a longer branch signifies that it took longer to evolve from its parent. So far, we saw the several algorithms to construct phylogenetic trees. Now, how do we know that the constructed tree is accurate? So we need a method to test its significance or confidence. So bootstrapping is an approach used to assess the reliability or significance or confidence of sequence-based phylogeny. Bootstrap values in a phylogenetic tree indicate that out of 100, how many times the same branch is observed when repeating the generation of a phylogenetic tree on a resampled set of data. It tells the confidence with which that branch occurs. Higher the value, higher is the significance. So, to do this, the sequence is randomly sampled from the original sequence, and a tree is constructed using the new sequence, and its similarity for each branch with the original tree is computed. So, this is done about 100 times, and the average confidence percentage value is considered to be the bootstrap value. So here, for example, in this tree, we could see the values here for each branch. So these are the bootstrap values. So a bootstrap value of more than 90 is considered to be of high confidence, and a value less than 50 is considered to be less confident. So this is used to assess the significance or the confidence of a constructed phylogenetic tree. Now, coming back to our question, which one of the following methods is used to test the significance of a predicted phylogeny? So, the answer would be option A, bootstrap, because that is the method used to test the confidence or the significance of constructed phylogenetic tree. So, maximum likelihood, maximum parsimony, and minimum evolution are the algorithms or the methods used to construct phylogenetic trees, as we discussed in the video. Thank you.